All right, folks, here we go, 24-5. So this is the stomach, and this is really just anatomy that you'll cover in lab. I'll point a couple things out, but the rest I'll do in lab. The alimentary canal has those four tunics I mentioned, and the muscularis externa for most of the alimentary canal is two layers, the circular layer and the longitudinal layer. However, the stomach has three layers. The stomach has an oblique layer. So you go mucosa, submucosa, muscularis externa. But in the muscularis externa of the stomach, we have an oblique layer, then the circular layer and the longitudinal layer, and then the serosa. So the muscularis externa is a little bit different in the stomach. It has the extra oblique layer. The most internal layer is the oblique layer. Remember that I told you, oh, maybe I didn't tell you. Actually, I don't think I did. The junction of the esophagus and stomach right here is called wow. It's called, well, some people call it the cardiac sphincter. Cardiac sphincter. Gastroesophageal sphincter. Gastroesophageal sphincter. Now, this is not a real sphincter. And when I say it's not a real sphincter, what I mean is anatomically, it's not a real sphincter. Anatomically, physiologically, it's a sphincter. Physiologically, it closes off. It keeps you from getting heartburn, also known as gastroesophageal reflux disease. Well, it's supposed to anyway. So physiologically, this can close off. It's a sphincter physiologically. But anatomically, it is not a discrete circular muscle that most sphincters are. It's not. However, the sphincter between the stomach and the duodenum right here, the pyloric sphincter, that is a true sphincter. That's a true anatomical sphincter. All right. The rest of this is really anatomy that we'll cover in anatomy. And what I really want to cover about the stomach is control and making secretions. So this is what the stomach looks like. You have these gastric pits, pretty deep gastric pits. You can see they go all the way down, and you have cells lining the pits. So these are gastric pits. You have the mucous neck cells in the neck. Those are mucous neck cells. You have actually more than two. You have several different types of cells inside these pits. You have to know what these cells do. The parietal cells make hydrochloric acid. The chief cells make pepsinogen. All right. So chief cells make pepsinogen, parietal cells make hydrochloric acid. G cells make gastrin. It's a hormone called gastrin. And you got to know what these guys make. Now we're going to talk about how the parietal cells make hydrochloric acid. But before we do, just let me tell you what hydrochloric acid does. The hydrochloric acid goes up through my pit and gets into my stomach, and it converts. Hydrochloric acid is necessary to convert pepsinogen into pepsin. Pepsin is an enzyme that digests proteins. So you're making the hydrochloric acid simply so you can convert pepsinogen into pepsin so you can digest proteins. So that's the story on that.
The stomach is located in the left upper quadrant of the abdominal cavity, between the esophagus and the duodenum, the first part of the small intestine. The function of the stomach is to mechanically and chemically process ingested food. Muscular contraction of the stomach wall and secretions from cells of the mucosa contribute to this function. The lesser curvature forms the superior border of the stomach, while the greater curvature forms the inferior border. Structurally, the stomach is divided into four regions. The cardia is a small region at the opening of the esophagus. The fundus is a dome-shaped area superior to the cardia. The body, the largest region, is located between the fundus and the pyloric part of the stomach. and the pyloric part is the narrow terminal portion between the fundus and the duodenum. The pyloric part is subdivided into the antrum, pyloric canal, and pylorus. The pylorus connects the stomach to the duodenum. It has a thick ring of smooth muscle called the pyloric sphincter that regulates the passage of stomach contents into the duodenum. Internally, the empty stomach is characterized by longitudinal ridges known as gastric folds or rugae. These folds allow the stomach to expand as it fills. The wall of the stomach has 4 layers: the serosa, muscularis, submucosa, and mucosa. The serosa is the thin outer layer composed of connective tissue. Unlike other parts of the digestive tract, the muscularis of the stomach is composed of 3 layers of smooth muscle: an inner oblique, a middle circular, and an outer longitudinal. These muscle layers assist in mixing stomach contents with gastric secretions and move food toward the pyloric sphincter. The submucosa is a connective tissue layer that attaches the muscularis to the mucosa. It contains blood vessels, nerve fibers, and lymphatics. The mucosa is composed of a simple columnar epithelium, a connective tissue layer known as lamina propria, and a smooth muscle layer called muscularis mucosae. Surface mucus cells in the epithelium secrete mucins, a component of mucus. Gastric mucus, which includes mucins and inorganic salts, protects the stomach lining from gastric secretions such as hydrochloric acid and caustic ingested compounds. Soft drinks, for example, have a pH ranging from 4.6 to 2.5. In addition, mucus also lubricates food for movement. The mucosa is indented by gastric pits that are lined with surface mucus cells. Branched tubular gastric glands empty into the base of the gastric pits. Gastric glands are composed of 4 cell types that produce specialized secretions involved in digestion. Mucous neck cells secrete mainly mucin. Parietal cells secrete hydrochloric acid and intrinsic factor. Chief cells, which are the most numerous cell type, secrete large quantities of the enzyme pepsinogen. Enteroendocrine cells secrete hormones and paracrine messengers that regulate digestion. Replacement of these secretory cells is provided by a population of rapidly dividing stem cells.
All right, folks, before I move on, I didn't mention in the previous slide that intrinsic factor is secreted by parietal cells. I failed to mention that. Let me just tell you something about intrinsic factor. It is a protein that's necessary for vitamin B12 absorption. You could eat all of the vitamin B12 in the world and not absorb any of it unless you have intrinsic factor secreted by parietal cells. And in fact, the deficiency of intrinsic factor is called pernicious anemia. And the treatment for pernicious anemia, and by the way, it's called anemia because you need vitamin B12 to make red blood cells. So without the vitamin B12, you don't make red blood cells. Without the red blood cells, you have anemia. The treatment for pernicious anemia is vitamin B12, but not dietarily. The way you get vitamin B12 to treat pernicious anemia is an intramuscular injection of it that bypasses the stomach. You don't have to worry about lacking intrinsic factor because you're not eating the B12, you're getting it injected into your muscle. So that's the intrinsic factor. And then the previous slide talked about G cells, and I said they secreted gastrin, this hormone called gastrin. And that's true, but what the video did was it classified all of the hormonal cells as enteroendocrine cells because more than just gastrin is secreted by some of those cells. There's other hormones as well. Now, just a reminder before we move on to making hydrochloric acid. This is a carbonic anhydrase reaction. Water plus CO2 gives us carbonic acid. This is carbonic acid. Like any acid, it dissociates into the proton and the conjugate base. All right, now strong acids dissociate almost fully. Carbonic acid is not a strong acid, but you still have this dissociation. By the way, this entire reaction is reversible. So I should have drawn these arrows double arrowed. This is a completely reversible reaction. That means bicarb and an acid can combine to make carbonic acid. Carbonic acid can dissociate into CO2 and water. This is completely reversible, this reaction. Now I want to talk to you about Le Chatelier's principle, the law of mass action. If I increase my substrates, it drives the reaction to the right. If I increase any of my products, it drives the reaction to the left. So this means a lot for a lot of different systems. For example, if I increase CO2 in my body, then I'm going to become more acidic. I'm going to drive the reaction to my right, and I'm going to increase my H pluses. I'm going to become more acidic. All right. If I become more acidic, I'm going to start driving, now not from increased CO2, but if I become more acidic for some other reason, so I become more acidic now first. I drive the reaction to the left, and I increase my CO2. Well, how do I get rid of the CO2? I can breathe it out. So by breathing out, I can get rid of some metabolic acids. Yeah, and that's why a lot of times you'll see diabetics that are in diabetic ketoacidosis. They do this deep, rapid breathing called Kussmaul respirations. What they're doing is they're blowing off the CO2 to try to adjust their pH back to normal by basically hyperventilating. So I can be breathing poorly, like chronic obstructive pulmonary disease or emphysema. I can breathe poorly and retain CO2 and become acidic. Or if I'm normally breathing and I have metabolic acidosis, I can increase my breathing and adjust my pH that way. So this is extremely important, this reaction. And the reason I bring it up in this chapter, by the way, you might remember that from 203. The reason I bring it up in this chapter is we're going to make hydrochloric acid here shortly. Well, we're going to show you how the stomach makes hydrochloric acid. Well, to make hydrochloric acid, you got to make the acid part. And then you got to have some chlorine and combine them, and you got hydrochloric acid. All right, so let's look at making hydrochloric acid. Before we
Cell membranes are lipid bilayers.
Sorry, I'm going to back that up. And before I play this, this is just to remind you about membrane transport. There's passive transport, there's active transport. It's been a while since you had 203, so let's just revisit this.
Membranes are lipid bilayers with charged groups on the surfaces and hydrophobic tails internally. With their hydrophobic cores and charged surfaces, cell membranes are very effective at blocking passage of many molecules. Some molecules, such as water, dissolved oxygen, and simple alcohols, are able to pass through cell membranes freely by passive diffusion. However, charged molecules and large molecules cannot pass directly through cell membranes, and hydrophobic molecules tend to become stuck inside the hydrophobic interior of cell membranes. In order to allow these complex molecules to enter and exit the cell, organisms rely on two forms of membrane transport: passive transport and active transport. Small uncharged molecules, such as dissolved oxygen, dissolved carbon dioxide, small alcohols, and small fatty acids can freely diffuse through the cytoplasmic membrane. They constantly diffuse back and forth in response to changing concentrations on either side of the membrane. Nonspecific facilitated diffusion is passive transport allowed by transmembrane proteins called permeases. In the case of nonspecific facilitated diffusion, these permeases allow a wide range of molecules that meet charge or size criteria to pass through their channel. Specific facilitated diffusion is passive transport allowed by transmembrane proteins called permeases. In the case of specific facilitated diffusion, these permeases only allow specific types of molecules to pass through their channel. The channel contains a recognition element. If the molecule is not recognized, the channel will not open to allow the molecule to pass into the cell. Osmosis is a special kind of passive diffusion that involves the movement of water molecules across a semi-permeable membrane. Water passes through a cytoplasmic membrane with some difficulty, but transmembrane channels can ease the passage of water through the membrane in a process called facilitated diffusion. Water plays a critical role in maintaining cell integrity. Water tends to move across the membrane from areas of high concentration to areas of low concentration, just like other solutes. The difference is that water makes up most of the liquid, so a change in the total concentration of solutes causes water to move. If the concentration of solutes outside the cell is high compared to the concentration inside the cell, water may leave the cell, causing the cell to shrivel up. Similarly, if the concentration of solutes inside the cell is high relative to the outside, water may move into the cell and cause it to swell up and possibly burst.
This discovery of water of osmosis actually being facilitated diffusion, because some simple diffusion occurs, but most of the water moves by facilitated diffusion. This discovery was by Peter Agre. He won the Nobel Prize for this, and these proteins right here that allow water through are collectively called aquaporins. Aquaporins. Now, there's a ton of aquaporins. When I say a ton, I know at least 4 of them. So I don't know if there's a ton. I don't even know if there's a dozen. I don't know how much a ton would be. They're just a protein. That'd be a lot of aquaporins. But these aquaporins, I'm trying to spell it better for you. These aquaporins, there's several different ones, and they all let water through, but they're coded for by different genes, and you'll find them in different tissues. That's the key. So Peter Agre, aquaporins.
A uniport is an active transport protein that transports a single molecule or a group of the same molecule in one direction against the concentration gradient. These proteins are also ATPases, using ATP to drive the transport. An antiport is an active transport protein that transports 2 molecules in opposite directions against their concentration gradients. These proteins are also ATPases, using ATP to drive the transport. An example of this kind of active transport protein is the sodium-potassium gated channels that maintain the electrochemical gradient of cells, which is responsible for maintaining a voltage difference between the outside and the inside of the cell.
Do you like how they call those pumps ATPases? Because they hydrolyze ATP into ADP plus inorganic phosphate. That's an enzymatic reaction, so they indeed are enzymes. They are transport proteins as well as enzymes. They are ATPases.
A symport is an active transport protein that transports 2 different molecules across the cell membrane at the same time. In this case, it is coupled with the active transport of a molecule against its concentration gradient performed by a uniport, which uses ATP to transport molecules against their concentration gradient. When the molecules are at a high concentration outside the cell, this concentration gradient can provide energy to transport the second molecule into the cell through the symport.
That's also called secondary active transport. If you look right here, this uniport right here just cleaved ATP. That's the source of energy for pumping this out. Now, what are these solutes typically? Sodium or protons. Sodium, they're typically either sodium or hydrogen ions. And then, let me play and stop again. Then what happens is the sodium or the proton comes back down its concentration gradient because you just pumped it all out. So there's a great concentration of these sodiums or protons on the outside. But they bring with it something going against its concentration gradient, this little square box. That could be a glucose or a sucrose or an amino acid. It could be any number of things. But it's called secondary active transport because this gray box right here is pumped against its concentration gradient, but there's no direct requirement for ATP. The energy comes from the concentration gradient of the sodium or the proton. But it's still active transport because how did the sodium and the protons get in such high concentration on the outside of the cell? The answer is ATP in the uniport system. So sometimes this coupled symport mechanism is also called secondary active transport.
Cell membrane.
All right, let's make some hydrochloric acid. This is the parietal cell right here in the stomach. So in the parietal cell, you have CO2 and water combined in the presence of carbonic anhydrase and make carbonic acid. Carbonic acid dissociates into a bicarb and a proton. We just showed you this when I showed you the carbonic acid reaction. Okay, I'm going to follow this bicarb in a second. All right. So then what happens? Let's do the proton first. Then what happens with the proton? Well, the proton is pumped into the stomach lumen. Now, what's not being shown here is this is an antiport. It's not showing it, and I wish it was. When the proton is pumped into the stomach, potassium is pumped into the cell. Now, the cell has a lot of potassium, so this is an active transporter, and it's an antiporter. Okay, now I have protons inside the stomach. Where's my chloride? I need hydrochloric acid. Let's go back. Chloride is a major extracellular anion. Here's chloride in the blood plasma. Okay, that's blood. What happens is, chloride being a negatively charged and bicarb being a negative charge, they do an antiport right here, or a counter-transport mechanism. The bicarb that came out of the carbonic anhydrase reaction enters the bloodstream, and the chloride enters the cell. By the way, no net change in charge, right? One negative left, one negative entered. Okay, let's look over here. Protons pumped into the stomach. Potassium's pumped into the cell. No net negative, no change in the charge of the cell. One positive was pumped out, one positive was pumped in. All right, now what happens here? The chloride, the chloride now is in high concentration inside the cell. The chloride diffuses into the stomach, and now I have hydrochloric acid. So that's how I make hydrochloric acid, right there. Now look at something else. When this bicarb enters the blood, it changes the pH of the blood right there. Now this is just a local pH change. Your overall body's pH of your blood is still 7.35 to 7.45. But the blood right past the stomach suffers from an alkaline tide because of all this bicarb entering the bloodstream as the blood flows past the stomach. And no problem, it'll be buffered, but there is an alkaline tide there. All right, so that we're going to look at this again with another picture, but that's how you make hydrochloric acid.
Hydrochloric acid is produced in parietal cells through a complex series of reactions. Carbon dioxide diffuses into the parietal cell, and the enzyme carbonic anhydrase catalyzes a reaction between the carbon dioxide and water to form carbonic acid. Carbonic acid dissociates into bicarbonate ion and hydrogen ion, and the bicarbonate ion is transported back into the bloodstream. An ion exchange molecule in the plasma membrane exchanges bicarbonate going out for chloride coming in. The hydrogen ions are actively transported into the duct of the gastric gland, and the negatively charged chloride ions diffuse with the positively charged hydrogen ions. Potassium ions are counter-transported into the parietal cell in exchange for hydrogen ions. The net result is production of hydrochloric acid in the parietal cells and its secretion into the duct of the gastric gland.
All right, that's making hydrochloric acid. All right, so let's talk about the three phases of gastric secretion. All right, and the three phases of gastric secretion are going to lead to the parietal cells making hydrochloric acid and lead to other cells telling whatever cells make that product to secrete their products, like the chief cell secreting pepsinogen and the mucous neck cell secreting mucus, and the enteroendocrine cells secreting gastrin and all of that. But we have three phases to this. So the three phases to gastric secretion, the first phase is the cephalic phase. This is the thought of food or the smell of food or the sight of food. This really has nothing to do with food on your tongue. All right, this is sight, smell, or taste, or even just the thought of food. What happens is your parasympathetic nervous system, in the form of the vagus nerve, fires, and it comes down to the stomach, and it says to the mucus cells, secrete mucus. It says to the chief cells, secrete pepsinogen. And it says to the parietal cells, secrete hydrochloric acid. And it says to the enteroendocrine cells, secrete gastrin. Now what gastrin's going to do is it's going to turn right back around and come back here and tell these very cells, mucus, chief, and parietal cells, secrete more of your products. But we're getting to that part. So the cephalic phase leads to all these secretions right here. And it also leads to your stomach churning, by the way. This is what happens when you're hungry and you smell food and your stomach just starts to churn. All right, that's the cephalic phase. The gastric phase. The gastric phase is when you put something into your stomach. So now you got food in your stomach. What happens? Your stomach distends. Your stretch receptors detect that. What happens is your stomach pH is usually around 1 or maybe 2, but it's pretty low. But when you put food into it, the pH rises because you're not eating food with a pH of 2. You're eating food with a pH of 6, so you make the pH of 2 of your stomach come up to 3 and 4 and maybe even 5. Well, the elevated pH is sensed by chemoreceptors. What happens? You have a local short reflex. Remember I told you in the two lectures ago, you're going to have these short reflexes and long reflexes. Well, this is a local short reflex. Stretch and elevated pH turn right back around on the stomach, and they say mucus cells secrete mucus, chief cells secrete pepsinogen, parietal cells secrete hydrochloric acid, G cells secrete gastrin. Meanwhile, the gastrin is carried by the bloodstream. It's a hormone. The gastrin comes right back and says to the chief cells, more pepsinogen, parietal cells, more hydrochloric acid. Now what's going to happen is pepsinogen, in the presence of hydrochloric acid, is going to become pepsin. Pepsin starts to digest protein. So protein digestion occurs in a great deal. In addition, my submucosal and my enteric plexuses are going to innervate the smooth muscles of the stomach, the three layers, the oblique layer, the circular layer, and the longitudinal layer, and you're going to churn the stomach. You're going to have mixing waves in the stomach. This is the gastric phase. And then you have the intestinal phase. Okay, now this gets a little bit tricky here. Overall, this duodenum down here, this duodenum needs to be basic, okay? So you can't put all of the stomach contents into the duodenum all at once, or you'll never neutralize it all. Well, first of all, the stomach can hold far more material than the duodenum. The stomach has all that rugae, and it can stretch and get quite big, filled with food. The stomach only gives little spurts of food to the duodenum at a time. Well, how is that possible? No problem. Watch. So a little bit of chyme from the stomach goes through this pyloric sphincter right here, and it gets into the duodenum. Now the stomach chyme is very acidic. This decrease in pH in my duodenum leads to secretin, a hormone to be secreted. Secretin goes up and inhibits the chief cells from making releasing pepsinogen, inhibits the parietal cells from making hydrochloric acid, and it inhibits peristalsis of the stomach muscles. All right? So now my stomach is slowing down. In addition, depending on the food I ate, if I ate lipids, CCK is secreted, and if I ate carbohydrates, gastroinhibitory peptide is secreted. By the way, I'm actually lying to you a little bit. Lipids and carbs can cause the release of CCK, and lipids and carbs can cause the release of gastric inhibitory peptide. I usually say lipids with CCK and carbs with GIP. It's not as specific as I'm making it sound. But the lipids and carbs cause the secretion of cholecystokinin. I better put that word here for you. CCK is called cholecystokinin. Cholecystokinin is a hormone, and gastroinhibitory peptide. Now, what does cholecystokinin and gastroinhibitory peptide do? Well, first of all, lots of things. But for right now, what we're going to say they do, and this is one thing they do, is they're going to feed up in the bloodstream. These are hormones. They're going to feed up to the chief cells and shut them down and shut down the parietal cells, just like secretin did. So the net effect here is slow down the stomach. When you first put your first bit of chyme in the duodenum, you slow down your stomach. All right. Let me see what else. In addition, the stretch, where am I right here? See right here? The stretch of the duodenum goes up and it shuts down the myenteric plexus to shut down the churning and pushing of my stomach food into my duodenum. This is called the enterogastric reflex. Do not confuse it with the gastroenteric reflex. There is a gastroenteric reflex, but I'm talking about the enterogastric reflex. Entero meaning intestine, gastric meaning stomach. The intestine to stomach reflex. When the duodenum fills with some food and the stretch and chemoreceptors are activated, I have the enterogastric reflex where I shut down the stomach. So these hormones and this enterogastric reflex, the hormones and the enterogastric reflex, lead to the stomach shutting down. Now, it's not permanent. The duodenum is going to do its deal and move the food along, move the chyme along, and then all this these inhibitory pathways will cease. And my stimulatory pathways will take over, and I'll start churning and put more chyme into my duodenum, and you'll do this whole thing again. So in other words, you don't empty the stomach all at once. You empty it a little bit at a time, and allow the duodenum to do its job. So this is the intestinal phase containing the enterogastric reflex. Now, do not confuse the enterogastric reflex with this reflex, the gastroenteric reflex, because here's what happens. The gastroenteric reflex is from stomach to intestine. When the stomach stretches, this dotted line, you can see the stomach stretching to the dotted line, it sends signals out to the intestines and it says move the food along. So intestinal motility increases. Peristalsis in my intestine increases. And right here's the duodenum, part of it. So the food moves from the duodenum into the ileum. I don't know what part of this is the ileum. Some part of this is the ileum. But the gastroenteric reflex increases the motility of the ileum that's the jejunum. Wow. It goes like this. It goes duodenum, jejunum, ileum. Anyway, you're moving food from the duodenum into the jejunum, from the jejunum into the ileum. Well, if you do this, you better open this ileocecal valve right here. The ileocecal valve is the valve between the ileum and the cecum. The cecum is the first part of the large intestine. No problem. When we do the gastroenteric reflex and move the food along, we also do the gastroileal reflex and we open that valve. So now the food is moving along out of the duodenum into the jejunum, out of the jejunum into the ileum, out of the ileum into the cecum because we just opened the valve. This moves the food along, and this is called the gastroenteric reflex of moving the food along. That's different from the enterogastric reflex where the intestines stop the stomach. All right, so that's the gastroenteric reflex and the gastroileal reflex. And this is the collage. And there you go. Play this over and over again if you're not getting it.
Regulation of stomach secretion is divided into 3 phases: cephalic, gastric, and intestinal. In the cephalic phase, the taste or smell of food, tactile sensation of food in the mouth, or thought of food sends nervous impulses to the medulla oblongata. These impulses cause parasympathetic neurons via the vagus nerves to stimulate secretion of hydrochloric acid and pepsin in the stomach. The parasympathetic stimulation also results in secretion of gastrin from the lower part of the stomach. This hormone travels through the bloodstream and further stimulates hydrochloric acid and pepsin secretion in the upper and middle parts of the stomach. In the gastric phase of gastric secretion, food has entered and distended the stomach. This distension activates a parasympathetic reflex via the medulla oblongata and also has a direct stimulatory effect on the gastric glands. The result is continued secretion of hydrochloric acid and pepsin. In the intestinal phase of gastric secretion, chyme has entered the duodenum, so gastric secretion is no longer needed. When the chyme contains lipids from the digestion of fats or contains enough hydrochloric acid to bring its pH below 2, gastric secretion is inhibited. The lipids and hydrogen ions inhibit gastric secretion by 3 simultaneous actions. First, they cause impulses to go to the medulla oblongata to decrease parasympathetic stimulation of the gastric glands. Secondly, they set up local reflexes via neurons in the wall of the gut that decrease gastric secretion. Thirdly, they cause the release of 3 local hormones which travel via the circulation to the gastric glands and inhibit their secretion.
All right. You can see the short reflexes and the long reflexes there, and the enteroendocrine reflexes there. You can see all of them.